when detailed knowledge would have been possible.
All right, let's start with the physical hardware itself. Let's dive into the core anatomy of the chromosome. Okay, let's unpack the basic structure. A chromosome isn't just a tightly wound piece of DNA, right? It's a complex molecular machine designed for packaging and access.
It is a machine, definitely. And its fundamental structure is called chromatin. Chromatin is simply the incredibly long DNA molecule wrapped around a specialized scaffolding of proteins known as histones.
Yeah, histones.
This packaging system is essential because human DNA, if you stretched it out, is about 6 feet long.
Wow.
Yet it must fit inside a microscopic nucleus. It's an incredible feat of engineering.
And we know exactly which histones are involved in that foundational packaging unit, the nucleosome.
We do. Histones H2A, H2B, H3, and H4 form an octamer. That's a set of 8 proteins, which acts as the core spool,
Like beads on a string.
exactly like beads on a string. The DNA wraps around this core about 1.65 times, creating the basic nucleus bead. Then, H1 acts like a specialized clamp, or linker, securing the DNA as it enters and exits the spool, helping to compact the structure further into what's called the 30-nanometer fiber.
And this winding process dictates whether a gene is accessible and active or silenced and packed away. It's not just storage.
No, it's active regulation, very active.
And when the cell decides to divide, that DNA must be duplicated first. Post-replication, after the S phase of the cell cycle, the duplicated DNA strand, still attached to its copy, is referred to as a chromatid.
Correct. When you see that classic X shape under a microscope, you are looking at 2 sister chromatids, identical copies held together at the center.
Okay, that X shape. So if that's the material, what are the structural landmarks? Every chromosome needs protection and a specialized way to move. What are the two key protective and functional structures?
That would be the centromere and the telomere. The centromere is that pinched-in region you mentioned, appearing sort of translucent in a stained karyotype. It's the attachment site for the specialized protein complex called the kinetochore.
Kinetochore, right.
This whole region is critically rich in repetitive DNA sequences, specifically alpha satellite DNA, and its job is singular. It is the precise docking station that ensures the chromosome can be grabbed and pulled correctly by the spindle fibers during division. It's like the handle.
Got it. And the telomere, the protective end caps. This is where the concept of the cellular clock comes in, which you said we'll delve into later.
Precisely. Telomeres are the physical protective tips at the very ends of the chromosome, defined by thousands of repetitive sequences. Specifically, the TTG repeats over and over again.
TTG.
Their function is vital. They act like the plastic tips on shoelaces, preventing the chromosomes from fraying, fusing with other chromosomes, or being mistaken for a broken piece of DNA that the cell should immediately try to repair.
And why do they act as a cellular clock? What's the mechanism there?
Well, it's because of the mechanism of DNA replication itself. The enzyme that copies DNA polymerase, cannot fully copy the very end of the lagging strand. There's just no place for the primer to attach right at the tip.
Okay.
Therefore, every time a somatic cell divides, a small segment of the telomere sequence is lost. It gets shorter and shorter.
Like a countdown timer.
Exactly. When they become critically short, the cell recognizes this as potentially dangerous damage and arrests the cell cycle, a process called senescence. That is the clock ticking down the lifespan of the cell.
Fascinating. And if we look at the complete organized set of these specialized structures, that defines the organizational chart of a species. We call this the karyotype.
The karyotype is the species-specific chromosome complement. It's an organized visual display where the chromosomes are stained, photographed, and arranged in homologous pairs, ordered by size from largest, chromosome 1, to smallest, chromosome 22, plus the sex chromosomes.
Right.
For us as humans, the characteristic karyotype is 46 chromosomes, arranged in 23 pairs. This visual map is absolutely foundational for diagnosing genetic diseases.
That perfectly sets the stage for our next section, which is about the spectacular dance of diversity, meaning meiosis, and how that precise mechanical process creates genetic variation.
The overarching goal of meiosis is deeply complex and twofold. First, the precise segregation of replicated chromosomes to generate haploid gametes, sperm, and egg cells, which is a reduction division. Cut the number in half.
Right, got to get down to half.
But perhaps even more fascinating is the second goal, simultaneously promoting maximum genetic recombination and variation. Diversity is the key output of this process. Evolution loves diversity.
Here's where the architectural changes begin. We start in prophase psi, where homologous chromosomes, the set inherited from mom and the set inherited from dad, pair up. That specific pairing process is called synapsis. forming structures known as tetrads because they consist of 4 chromatids.
And that pairing isn't accidental. It's meticulously controlled by a massive, central, zipper-like protein structure called the synaptonable complex.
A zipper. Okay.
This complex aligns the homologs base pair by base pair across their entire length, ensuring precise registration so that the critical step of crossing over can occur without catastrophic errors. It holds them perfectly still.
Crossing over. Okay, this is the big one for variation, right? This is the genetic exchange between non-sister chromatids, the shuffling of maternal and paternal DNA, and the visible physical location where this exchange happens is called the chiasma or chiasmata, plural. This is the primary process that shuffles the deck and generates new, diverse combinations of alleles.
Yes, and the mechanism is really sophisticated. Crossing over is initiated by the enzyme SPOA1, which intentionally induces double-strand breaks in the DNA.
Intentionally breaks the DNA.
Yes. Think of it as a carefully controlled suicide mission for the DNA strand, necessary to initiate the repair and exchange process.
That sounds dangerous, intentionally breaking the DNA backbone. What prevents the cell from just panicking and trying to, patch it up immediately?
The machinery is very tightly controlled. Once broken, specialized recombinase enzymes, notably RED51 and DMC1, take over. They invade the homologous chromosome, scan for homology, find the matching sequence, and then physically guide the repair process to exchange the DNA segments between the non-sister chromatids.
So it uses the other chromosome as a template, but also swaps pieces?
Exactly. This exchange is the very definition of genetic variation, creating chromosomes that are unique blends of the 2 original parental copies. You get mosaic chromosomes.
Once that intricate shuffling is done, we move to metaphase Earth, which provides the second major mechanism for diversity, independent assortment.
Right. The key here is the positioning of the tetrads, those paired homologous chromosomes. They line up randomly along the metaphase plate, the equator of the cell.
Completely random.
Completely random. Imagine flipping a coin for every single one of the 23 pairs. The orientation of chromosome 1, whether the mom's copy points north or south, has absolutely no bearing on the orientation of chromosome 21. This random alignment ensures that when they separate, the resulting gametes possess a massive exponential number of possible combinations. 2 to the power of 23, basically.
That's a huge number.
It's enormous.
Yeah.
Millions of possibilities just from assortment alone.
And in anaphase for Hurst, the separation happens. The homologous chromosomes are pulled apart towards opposite poles, powered by spindle microtubules using kinesin and dynein motor proteins. But here is that crucial mechanical distinction between meiosis the first and meiosis the second, which determines if the division is successful.
Yes, the distinction lies entirely in the protection and degradation of the protein cohesin. Cohesin is the molecular glue that holds the 2 sister chromatids together.
Okay, the glue.
In anaphase the nice, the enzyme separase is activated, which degrades cohesin along the arms of the chromosome. This allows the homologs, the 2 different X's in the tetrad, to separate.
But the centromeric cohesin, the glue right at the center, must be protected.
Absolutely vital.
Yeah.
Cohesin at the centromere must be shielded by another protein, Shugoshin, until meiosis the second. If the sister chromatids separated in meiosis the first, the whole division would fail, resulting in aneuploidy, the wrong chromosome number.
So meiosis the second is essentially like a modified mitotic division then?
Correct. It proceeds rapidly without further DNA replication. Now, in anaphase second, the remaining centromeric cohesin is finally degraded, the sister chromatids separate, and the process results in 4 unique haploid cells ready for fertilization. The control of that cohesin degradation, when and where it happens, is the mechanical genius of meiosis.
That's the mechanism for segregation and diversity. But sometimes genes don't follow the rules of independent assortment. They seem to stick together. They're inherited together. This brings us to the concept of linked genes.
Exactly. Genes located physically close together on the same chromosome are said to be linked. They are generally inherited together as a block, unless they are separated by the random crossing over event we just discussed.
So the closer they are.
The closer the 2 genes are on the chromosome, the less likely a crossing over event will occur between them. They tend to travel together.
And that separation likelihood can be quantified using something called the recombination frequency. How does that work?
Precisely. We calculate this frequency by counting the number of recombinant offspring, those who show a shuffled combination of parental traits, different from either parent, and dividing that by the total number of offspring, then multiplying by 100.
Okay, so it's a percentage.
It's a percentage, and this percentage gives us a map unit called a centimorgan, or cm. If 2 genes are 5 centimems apart, it means they are separated by crossing over 5% of the time. If they're 50 cm apart, they essentially assort independently, behaving as if they were on separate chromosomes because crossing over happens between them so often.
Yeah, so you can build maps just based on how often traits recombine.
Exactly. This measurement allowed scientists like Morgan and his students to build detailed, accurate genetic maps decades before sequencing was commonplace. It was a landmark achievement.
If we connect this quantitative knowledge to the bigger picture, this non-random association of alleles is central to modern genomics research, isn't it? This idea of linkage.
Absolutely. This persistence of linked blocks is what we call linkage disequilibrium, LD. LD is the non-random association of alleles at different locations across the genome. If a beneficial or maybe a disease-causing mutation arose thousands of years ago on a particular chromosome, the region surrounding it, known as the haplotype block, remains linked over many generations because crossing over rarely breaks up very large sections completely, especially if the points are close.
And this LD is why modern genome-wide association studies, GWAS, are possible. It's a shortcut.
It is the ultimate shortcut. We don't have to sequence every single base pair for every person in a huge study, which would be incredibly expensive and slow.
Right.
Instead, researchers only need to track a few key markers, called single nucleotide polymorphisms, SNPs, that are situated within a block of LD. If that marker SNP is statistically associated with a disease,
Then the actual gene causing it is probably nearby.
exactly, we know the actual causative gene is nearby, somewhere within that same linked block, then you can zoom in and sequence just that region.
That is a powerful connection, showing how the physical constraints of chromosome movement directly dictates our ability to perform cutting-edge medical research and find disease genes. Amazing.
But of course, now we need to explore what happens when this precise chromosomal dance goes wrong.
Let's discuss when things go wrong, alterations and aneuploidy. When chromosomes fail to segregate properly, this is called non-disjunction. This failure is the cause of aneuploidy, where an individual has an abnormal number of chromosomes.
Non-disjunction is highly sensitive to timing, as we hinted before. It can occur in meiosis the first, where the homologous chromosomes fail to separate.
So one cell gets both, the other gets none.
Right. Or it can occur in meiosis the second, where the sister chromatids fail to separate. Either way, the resulting gametes will carry an incorrect number of chromosomes, leading to a zygote with a potentially lethal imbalance after fertilization.
And the most common survivable type is trisomy, 2n+1, an extra chromosome.
Trisomies often result in conditions we are very familiar with, probably the most well-known being Down syndrome, which is trisomy 2131, 3 copies of chromosome 21. While we survive trisomy 21, the gene dosage imbalance leads to recognizable features, cognitive delays, and specific health issues like congenital heart defects. Other, much more severe trisomies include Edwards syndrome, trisomy 18, and Patau syndrome, trisomy 13, which are often lethal shortly after birth due to the magnitude of the genetic imbalance. Too many genes are affected.
And regarding the sex chromosomes, we see survivable conditions like Klinefelter syndrome, XXY, where a male has an extra X chromosome, leading to reduced fertility and sometimes mild developmental effects.
Correct. Conversely, if a chromosome is missing, that's monosomy, 2n1. Autosomal monosomies, missing non-sex chromosomes, are almost universally lethal very early in development, suggesting the loss of even one copy of those essential genes is incompatible with life.
So you need 2 copies of most chromosomes.
Generally, yes. The most common survival monosomy involves the sex chromosomes, Turner syndrome, XO, where a female has only a single X chromosome, resulting in short stature and infertility, among other things.
And just for completeness, we should mention polyploidy having entire extra sets of chromosomes like triploidy, 3n, or tetraploidy, 4n. That sounds pretty drastic.
It is, for humans. While polyploidy in humans is usually lethal and results in spontaneous miscarriage, the plant kingdom is much more tolerant.
Ah, interesting.
As the source notes, polyploidy is frequently exploited in agriculture because it often results in larger, more vigorous plants or sterile traits, such as the triploid bananas and seedless watermelons we enjoy. It's an interesting contrast in genetic tolerance between different forms of life.
Definitely. Beyond the sheer number, we also have to consider structural alterations changes to the physical arrangement of the genes within the existing chromosomes. Let's run through those structural changes because they have very specific molecular and clinical consequences.
Okay, if a segment is lost, that's a deletion. The molecular consequence of a deletion is often haploinsufficiency, meaning having only one functional copy of a gene isn't sufficient for normal function, leading to basically a half dose of the required protein.
And what's the classic example there?
The classic example is Cri du Chat syndrome. caused by a deletion on the short arm of chromosome 5, designated 5p deletion. Clinically, this leads to microcephaly, profound intellectual disability, and the distinct, high-pitched cat-like cry in infancy due to laryngeal abnormalities. Another severe example is the deletion associated with retinoblastoma 13q14.
Okay. And the opposite side of that coin is a duplication, where a segment is repeated.
Duplication causes a gene dosage effect. Instead of having too little protein, the cell produces too much of the protein coded by that repeated segment. Even minor dosage imbalances can disrupt complex developmental pathways.
So too much can be just as bad as too little.
Often, yes. For example, Charcot-Marie-Tooth disease, a peripheral neuropathy, is often caused by a duplication of the PMP22 gene on chromosome 17, leading to progressive muscle weakness.
Then there's rearrangement. If a segment reverses its orientation, gets flipped 180 degrees, that's an inversion.
Inversions can be tricky. A person carrying a balanced inversion might be phenotypically healthy because all their genes are still present, just rearranged.
Right. Nothing lost or gained.
Exactly. However, during meiosis, the inverted chromosome must try to pair with its non-inverted homologue, leading to the formation of an unusual loop structure. This can suppress recombination in that area and often leads to problems in gamete formation, unbalanced chromosomes in sperm or egg, resulting in infertility or miscarriages.
Ah, okay. The most famous structural alteration, especially in the context of cancer, must be the translocation, where a segment moves to a non-homologous chromosome.
Absolutely. Translocations are critical because they often create novel, hybrid genes or move genes into regions where they are inappropriately expressed. The poster child for this is chronic myelogenous leukemia, CML.
Right.
This cancer is defined by the Philadelphia chromosome, which results from a reciprocal translocation between chromosomes 9 and 22, designated T922. They swap pieces.
And what's the outcome of that fusion? What does that specific swap do?
The translocation creates the BCR-ABL fusion protein. The ABL gene, which codes for a tyrosine kinase involved in cell growth signaling, is normally tightly regulated. But when it fuses with the BCR gene on chromosome 22, it creates a hyperactive, always-on tyrosine kinase. Always on. This runaway signaling protein drives uncontrolled cell growth and division, making it the perfect, specific molecular target for modern chemotherapy drugs like Gleevec, which specifically inhibit BCR-ABL.
Incredible example of targeted therapy based on a chromosomal flaw. We should also briefly note the mechanisms behind some of these chromosomal mutations, tying back to McClintock's work. Transposons, the mobile DNA elements.
Yes, they are powerful drivers of genomic change because they are literally jumping genes that can copy or move themselves, potentially disrupting coding regions, splicing sites, or regulatory sequences when they land somewhere new. They're classified by how they move.
What are the two main classes?
We have class 1, or retrotransposons. These elements are copy and paste. They first transcribe themselves into an RNA intermediary and then use an enzyme called reverse transcriptase like retroviruses use to create a new DNA copy that inserts itself elsewhere in the genome. So the original stays and a new copy appears.
Okay, copy-paste.
Then we have class 2, or DNA transposons. These are cut and paste elements. They excise themselves directly from their original location and reinsert into a new one using an enzyme called transposase. The original copy is gone from its first spot.
Got it. And finally, large-scale gains or losses of DNA covering kilobases to megabases are broadly termed copy number variations, or CNVs. These are bigger than small insertions deletions, but may be smaller than whole chromosome aneuploidy.
Exactly. CNVs are incredibly important in fields like neurodevelopmental research. These large structural variants disrupt the genome by altering gene copy number and changing chromatin architecture over a significant region. For instance, deletion or duplication in the 16p11.2 region is one of the strongest genetic risk factors currently identified for conditions like autism and schizophrenia.
Wow.
Finding these CNVs using techniques like arrays, which we'll discuss, is now a critical part of diagnosis for many developmental disorders.
That transition from physical structure and gross errors now leads us beautifully to Section 4, beyond the sequence. We're now exploring mechanisms of heredity that regulate gene expression without altering the underlying DNA sequence, starting with sex determination.
Right. The determination of sex in mammals is anchored by the X and Y chromosomes.
Right.
XY means male and XX means female. This is determined almost entirely by the presence or absence of a single crucial gene on the Y chromosome, the SRY gene.
SRY. It's the master switch that sets development down the male pathway.
Exactly. The SRY gene produces a transcription factor, a protein that turns other genes on or off. This factor triggers the differentiation of the initially indifferent gonad into the testes. Once the testes forms, it produces the hormones, like testosterone, that drive subsequent male development. Without SRY, the default pathway is female.
Because males have only one X chromosome, their XY, they are much more susceptible to traits carried on it. This is sex linkage.
Correct. X-linked recessive traits are expressed much more frequently in males because they are homozygous for that chromosome. They have only one copy. They lack a second X chromosome to provide a healthy dominant allele to mask the recessive one if they inherit it.
Right.
This is why classic examples like hemophilia A, a blood clotting disorder, and certain types of red-green color blindness disproportionately affect males.
Okay, so if males only have one functional X chromosome's worth of genes active, why do females with two X chromosomes not produce double the required X-linked proteins? That seems like it would cause a huge imbalance, which brings us to dosage compensation.
Dosage compensation is absolutely essential for balancing gene output between the sexes. In females, one X chromosome is randomly inactivated very early in embryonic development, ensuring that both males and females have roughly equivalent amounts of X-linked gene product.
Randomly inactivated.
Yes, random in each cell lineage. The inactive X chromosome condenses into a tightly packed, transcriptionally inactive structure known as a bar body. You can sometimes see it under a microscope as a dense spot near the nuclear envelope.
And what mediates this silencing? How does the cell pick one X and shut it down?
The process is mediated by a specialized non-coding RNA called ISPS RNA. ISPS RNA is transcribed from the X chromosome that will become inactive. It then coats that same chromosome.
Ah, coats it.
Yes, it physically spreads along the chromosome, leading to massive shifts in histone modification, like methylation and deacetylation, and DNA methylation that effectively lock the entire chromosome down, silencing most of its genes.
And the beautiful visible result of this random inactivation, or mosaicism, is seen in the coat pattern of calico cats.
A perfect, easily visualized genetic phenomenon. Since the gene for coat color, orange versus black, is X-linked in cats, different patches of skin cells randomly inactivate either the paternal X or the maternal X. If the cat is heterozygous for the color alleles, this leads to the characteristic mosaic pattern of orange and black fur patches. White patches are due to a separate gene.
Very cool. From the visible mosaicism to the microscopic switches that control which genes are silenced, let's talk about epigenetics' heritable changes in gene expression without altering the underlying DNA sequence. This is the ultimate regulatory layer, isn't it?
It really is. Epigenetics determines which genes are expressed when, where, and how strongly, and it's influenced by everything from development to diet to environment. There are three primary mechanisms we need to focus on, and they all work together to control the packaging of the chromatin. First, DNA methylation.
Okay, methylation. This involves the addition of a methyl group to specific cytosine bases. Where does that typically happen?
Primarily in specific sequences where a cytosine C is followed by a guanine G, creating CpG islands. These CpG islands are often found clustered in the promoter regions of genes, the on-off switch areas.
And what does adding that methyl group do?
Adding that methyl group generally acts as a signal to science promoters and condense the surrounding chromatin. Methylated DNA attracts proteins that help compact the chromatin, effectively locking the gene away from the transcriptional machinery, turning it off.
How is this critical pattern maintained when the cell divides? Because the daughter cells need the same identity, right?
That's a crucial point. This is where specialized enzymes come in. Enzymes like DNMT3A and DNMT3B establish new methylation marks, de novo methylation, during development. But importantly, the pattern is maintained through cell division by DNMT1.
DNMT1.
DNMT1 recognizes the existing methylation marks on the parental DNA strand after replication and places identical marks on the newly synthesized daughter strand. This ensures that a silenced gene in a liver cell stays silenced in all subsequent liver cells, maintaining cell identity.
Okay, that's methylation. The second major mechanism involves those histone proteins again. Histone modification.
Right. Since DNA is wrapped around histones, chemically modifying the histone tails, the parts that stick out from the core, changes how tightly the DNA is packed. This is often called the histone code.
So what kind of modifications are we talking about?
A key one is acetylation. The addition of an acetyl group to lysine residues on the histone tails neutralizes their positive charge. Since DNA is negatively charged, this weakens the histone's grip on the DNA.
It loosens it up.
Exactly. This leads to open chromatin, often called euchromatin, meaning the genes in that area are active and accessible for transcription.
And the reverse process to shut things down.
The reverse process deacetylation, removing acetyl groups, and specific types of histone methylation, Adding methyl groups to histones, which is more complex than DNA methylation, can increase the positive charge or recruit silencing proteins, tightening the packaging. This leads to condensed chromatin, or heterochromatin, and gene repression. The dynamic balance of acetylation and methylation is key to controlling gene activity moment by moment.
It sounds incredibly complex, like a whole other layer of information on top of the DNA sequence.
It absolutely is.
And the clinical relevance of epigenetics is starkly seen in imprinting, where a gene's expression depends entirely on whether it was inherited from the mother or the father.
Imprinting is fascinating because the epigenetic tag, usually a methylation mark, is placed on the gene during gamete formation, in the sperm or the egg, and somehow survives fertilization and reprogramming. The resulting child only expresses the gene copy from one specific parent, even though they possess two copies in their DNA sequence. The other copy is silenced by the imprint.
How does that manifest clinically? Give us an example.
The source gives a classic, striking example involving chromosome 15. If a segment of chromosome 15q is lost, a deletion, the resulting disorder depends purely on the parent of origin of the remaining chromosome.
Okay.
If the deletion is on the paternal copy and the maternal copy carries genes that were epigenetically silenced, imprinted during egg formation, you get Prader-Willi syndrome, characterized by obesity, intellectual disability, short stature.
Right.
Conversely, if the deletion is on the maternal copy and the paternal copy has different genes in that region silenced during sperm formation, you get Angelman syndrome, characterized by severe intellectual disability, seizures, jerky movements. Same genomic deletion location, completely different severe clinical outcomes, dictated purely by the parental origin epigenetic tag.
That's incredible. Same dilution, different diseases based on which parent it came from.
Precisely. It highlights the power of epigenetic regulation.
Finally, let's look at inheritance outside the nucleus. Mitochondrial inheritance, this is a bit different, right?
Yes, this is the critical exception to standard Mendelian nuclear inheritance rules. Mitochondria, the powerhouses of the cell, contain their own small genome. Since the sperm contributes essentially only its nucleus to the zygote, and its mitochondria are generally destroyed after fertilization.
All the mitochondria come from the egg.
Correct. All mitochondrial DNA, or mtDNA, is inherited solely from the mother. It's a purely maternal inheritance pattern.
And the mtDNA is quite small, but functionally mighty. What's on it?
It's small circular DNA, typically containing 37 genes in humans. These genes are vital, coding for 13 essential proteins involved in the electron transport chain, ETC, the engine of ATP energy production, plus 22 tRNAs and 2 RNAs needed for protein synthesis within the mitochondrion itself. They build parts of their own machinery.
Why does this separate genome tend to cause disease? You hear about mitochondrial diseases.
Because mtDNA lacks the protective packaging of histones found in the nucleus, and it's constantly exposed to highly damaging reactive oxygen species, ROS, generated as a byproduct of the ETC running right there.
Ah, it's right in the line of fire.
Exactly. As a result, it has a mutation rate significantly higher than nuclear DNA.
Yeah.
These mutations primarily affect the ETC subunits, leading to ATP deficiency, since mitochondria power high-energy organs like muscles, brain, eyes, and heart.
Those are the organs most affected?
Precisely. The resulting disorders are typically devastating neuromuscular diseases. Examples include Leber's hereditary optic neuropathy, LHON, which causes blindness, MELOS, mitochondrial encephalopathy, lactic acidosis, and stroke-like episodes, and MRF, myoclonic epilepsy with ragged red fibers, all inherited maternally.
Okay. Now that we've established the structure, the mechanics, the errors, and the regulatory systems like epigenetics and mitochondrial inheritance, let's move to our final section. Precision diagnosis, applying chromosomal knowledge in the clinic.
Right. What's fascinating here is realizing that chromosomes are not just linear sequences, they are self-organizing 3D networks within the nucleus. The way they fold and interact spatially matters for gene regulation and overall cell identity. When these networks are fundamentally disrupted by translocations, large CNVs, or catastrophic epigenetic changes, we get disease, especially cancer, which often features chaotic genomes.
And connecting back to those protective caps, telomere shortening, we mentioned acts as a clock. How does that play into disease and health?
Telomere shortening acts as a definitive cellular clock, as we said. When telomeres reach a critically short length, they trigger DNA damage signals, leading to senescence. an irreversible cell cycle arrest.
So the cells just stops dividing.
It stops dividing. This is thought to be evolution's built-in tumor suppressive barrier, halting uncontrolled growth of damaged or old cells before it can start. To become cancerous and divide indefinitely, most cancer cells must find ways to overcome this barrier, typically by reactivating the enzyme telomerase to elongate their telomeres again, effectively making them immortal.
Counteracting the clock.
Exactly. Targeting telomerase is actually a strategy in cancer therapy development.
Okay, so modern medicine relies on techniques that can diagnose these chromosomal problems at every level of resolution. How do doctors actually detect these changes? It depends on the scale of the problem, doesn't it?
It does. We start with the most basic, lowest resolution tool, the karyotype.
The picture of the chromosomes we talked about earlier.
Exactly. Karyotyping involves growing cells, arresting them in metaphase, staining them, usually with G-banding to get a distinct banding pattern, and arranging the images. It provides relatively low resolution detecting changes, typically larger than 5 to 10 million base pairs. But it's excellent for the initial screen. It detects gross changes, primarily the aneuploidies we discussed, like confirming Down syndrome, trisomy 21, Turner XO, Klinefelter XXY, or identifying very large structural rearrangements like the Philadelphia chromosome in CML.
But if we suspect a smaller deletion or duplication in a specific known location, maybe associated with a known syndrome, we need to focus in. We move to a targeted approach using FISH.
Right. FISH, or fluorescent in situ hybridization, uses fluorescently labeled DNA probes designed to hybridize specifically to certain loci or even entire chromosomes.
So you use a glowing tag for a specific gene or region?
Precisely. This technique provides medium resolution down to about 100 kilobases, kbb, up to 1 millib. It's invaluable for confirming specific microdeletion syndromes, like Creux-Duchat or DeGeorge syndrome, or translocations that are too small to visualize reliably with standard karyotyping. You're looking for the presence, absence, or wrong location of specific colored fluorescent signals under the microscope.
But what if you suspect a small deletion or duplication, like one of those CNVs involved in developmental delay, but you have no idea exactly where in the 46 chromosomes to look? That's where you need a whole genome survey, but with better resolution than a karyotype.
Then we move to array CGH, or comparative genomic hybridization on a microarray. This is a high-resolution technique, capable of detecting sub-megabase CNVs across the entire genome in one go, often down to 50 kilobase or even smaller.
How does it work? Comparing.
It works by comparing the patient's DNA signal against a control reference, DNA sample. Both DNAs are labeled with different fluorescent dyes, say red for patient, green for control, and hybridized to a chip covered in probes representing the entire genome. If the patient DNA signal is brighter, more red in a region, it's a duplication, again. If it's dimmer, more green, it's a deletion, a loss. If it's yellow, equal red and green, it's normal copy number.
Okay, so it scans everything for gains and losses.
Yes, it's the standard workhorse now for finding small copy number gains or losses, especially in cases of unexplained developmental delay or intellectual disability where a specific syndrome isn't obvious.
What's the major limitation of array CGH, though?
The crucial limitation is that array CGH cannot detect balanced rearrangements, like balanced translocations or inversions. Since a balanced rearrangement involves no net gain or loss of DNA, it's just rearranged, the comparative array will register a normal signal, yellow. It can't see that pieces are in the wrong place, even if those rearrangements are causing infertility or risk of unbalanced gametes.
Right, because the total amount of DNA is the same. So for the ultimate level of detail, especially in cancer genetics where things get really complex, or for finding those balanced rearrangements, we have to look at the highest resolution. Whole genome sequencing, WGS.
Exactly. WGS offers base pair resolution. This is the comprehensive analysis that theoretically detects all types of variants. Not only does it find the small sequence changes, like single nucleotide variants or SNPs, but critically, it also detects CNVs and precisely identifies the exact breakpoints of complex structural rearrangements, including balanced ones.
So you see exactly where the break happened and where the pieces rejoin.
Precisely. For a complex cancer like CML, WGS confirms the exact base pair fusion point of the BCR-ABL gene, providing the most precise diagnosis possible for targeted therapy. Or it can find novel gene fusions driving other cancers. It's becoming more common, though still more expensive and complex to analyze than arrays.
So the combination of cytology, The study of the physical chromosomes and molecular genomics, the sequencing of their DNA is truly driving precision diagnosis and the development of targeted therapies based on pinpointing the exact molecular failure.
It really allows us to move beyond just treating symptoms and actually target the fundamental cause, whether it's a non-disjunction event leading to Down syndrome that needs specific management or a fusion protein driving a specific leukemia that needs a specific inhibitor drug.
It's truly personalized medicine based on the chromosomal blueprint.
Absolutely. So we have covered vast ground today, mapping the chromosomal blueprint of life. Modern heredity, as you now know, is not just abstract rules like Mendel figured out. It's a complex interplay of physical movement during meiosis, dynamic regulatory switches in epigenetics, and even specialized inheritance through the mitochondria. Cytogenetics combined with molecular genomics now allows for this amazing precision diagnosis and targeted cancer therapies.
Yeah, I think the key takeaway for you listening is that chromosomes aren't just passive carriers of DNA. They are dynamic, self-organizing networks whose precise 3D structure and folding dictates when and where genes are active. They are the ultimate physical realization of Mendel's laws, but way more complex and dynamic than he could have imagined.
Well put. And this knowledge gives us profound insights into human health and disease. We've discussed telomeres as a cellular clock that stops growth, initiating senescence, acting as a crucial tumor suppressive barrier. But this raises an important, maybe slightly unsettling question for you to mull over.
Okay.
What are the implications when cancer cells reactivate the ability to elongate their telomeres using the enzyme telomerase, essentially making their cellular clock run backward and achieving a form of biological immortality? If the physical limits of cell division can be broken by reactivating this enzyme, what does that mean for the biology of aging itself, and could we or should we ever try to harness that?